So my name is Bernardo Trigatti. I go by Dino, so please feel free to call me Dino. I'm a professor in the Department of Biochemistry and Biomedical Sciences at McMaster University, and I'm a scientist and researcher in the Thrombosis and Atherosclerosis Research Institute that's affiliated with both McMaster University and Hamilton Health Sciences. And my research focus is on molecular mechanisms of atherosclerosis development. So what we're interested in are what are the biochemical and genetic pathways that are involved in development of atherosclerosis, and particularly are there pathways that are new and previously unrecognized that we can target with therapeutic drugs as potential pathways to prevent or reverse atherosclerosis development. Okay, so atherosclerosis is commonly called hardening of the arteries. And basically what it is a condition where there's an accumulation of cholesterol in the walls of arteries that causes the artery walls to thicken. And this narrows the size of the artery, of the lumen of the artery through which blood can flow. So it narrows the diameter of the artery and restricts blood flow to tissues. And when this happens in arteries that feed the heart, this restricts blood flow to the heart, causes the heart muscle cells to become starved of oxygen and nutrients, and essentially triggers a heart attack. If this happens in arteries that feed the brain, the result is a stroke, an ischemic stroke. And if this happens in other arteries, peripheral arteries, the result is peripheral arterial disease. So this narrowing of the arteries because of buildup of cholesterol really involves a remodeling, a cellular remodeling of the makeup of the walls of the arteries. So there's an invasion of the walls of the arteries with cells that make up part of the innate immune system, inflammatory cells called macrophages. And these macrophages are the cells that accumulate these large amounts of cholesterol. So what we're trying to understand is what are the factors that trigger the influx of macrophages into the artery wall? And is this process reversible? Can we trigger movement of these cells out of the artery wall to try to reverse the disease? Well, I came at this actually through a background in studying a particular molecule called scavenger receptor class B type 1. It turns out that this molecule acts as a receptor for high-density lipoproteins. And high-density lipoproteins are complexes of cholesterol and protein that serve to carry cholesterol throughout the body. So these are lipoproteins in general. And they come in different types. So there's high-density lipoprotein, low-density lipoprotein, and certain other types. Now, high-density lipoprotein is commonly known as the good cholesterol. So cholesterol, having large amounts of cholesterol associated with high-density lipoprotein, is associated with reduced risk of atherosclerosis, whereas low-density lipoprotein, or LDL, is called the bad cholesterol because having large amounts of LDL cholesterol is associated with increased risk. So because I was studying this receptor, which turned out to be a receptor for high-density lipoproteins, we decided to test whether this receptor played a role in atherosclerosis by generating a knockout mouse that lack this receptor to see whether atherosclerosis development was impacted. And that's really how I got into the atherosclerosis field. One case where we did look at dietary supplementation on atherosclerosis development was a study that was led by a former PhD student of mine a number of years ago, and that is Dr. Aisha Al-Jarala, and she's now a professor at Kuwait University. And she was interested in the potential effect of antioxidants on development of atherosclerosis in a mouse model that we have that develops atherosclerosis in coronary arteries, and these mice also develop heart attacks as a result of it. So her interests were in seeing whether pomegranate extract could impact the development of atherosclerosis in coronary arteries and could impact the development of fibrosis or scarring in the heart that results, we think, from the mice developing heart attacks. And what she found was when she supplemented the drinking water, so it wasn't the diet per se, she kept the diet the same, but she put pomegranate extract in the drinking water of these mice. She saw reduced levels of atherosclerosis, reduced levels of coronary artery disease, and reduced scarring in the hearts, suggesting that the pomegranate extract was in fact protecting them from atherosclerosis and subsequent heart attacks. Well, we think what happened in that case is that the pomegranate extract is rich in antioxidants, and it prevented a process called development of reactive oxygen species. So one of the things that contributes to atherosclerosis is that there is production of these oxidative molecules, so-called reactive oxygen species, that themselves have a variety of effects. They alter how cells behave, the cells that make up the atherosclerotic plaque. They change the chemical nature of proteins. They trigger accumulation of lipoproteins in the artery wall, and act as inducers of these inflammatory cells. So they have a variety of negative effects. And what we think is happening, and we had some evidence in that paper, is that the supplementation of the drinking water with this pomegranate extract reduced the level of oxidative stress or the abundance of these reactive oxygen species molecules. And we think that this is the pathway by which it protected against atherosclerosis. It certainly wasn't, like, we didn't include enough of the pomegranate extract to really have any nutritional effect to the mice who were getting it. So we think that it was acting as a sort of like a micronutrient. So we're starting to do those types of experiments now. What we've become interested in regards to the scavenger receptor for HDL, the HDL receptor, is trying to understand its role in macrophages. So again, these are the inflammatory cells that move into the atherosclerotic plaque. And we're trying to understand its role in these cells as well as in cardiomyocytes, the heart muscle, the cells that make up the heart muscle, in protection of these cells against cell death during both atherosclerosis development in the case of macrophages, but also during myocardial infarction in the case of the heart muscle cells. And it turns out that high-density lipoproteins can act as, in a sense, like signaling molecules to trigger activation of cell signaling pathways in macrophages and in cardiomyocytes. And the HDL receptor plays an important role in both cell types in this. So not only does HDL work by sort of clearing cholesterol from the affected artery, it actually triggers changes in the cells that interact with HDL that make those cells more resistant to dying. And this helps in the case of an atherosclerotic plaque because it keeps the macrophages alive and allows them potentially to then be able to move out of the atherosclerotic plaque to trigger so-called plaque regression. And in the case of the heart muscle, of the cardiomyocytes, when these cells are starved of oxygen and nutrients, the cells themselves undergo cell death. And HDL, it turns out, protects them against that in a process that's dependent on the HDL receptor. And so if we can keep those heart muscle cells alive, then we can minimize the development of the myocardial infarction, of the heart attack. This receptor is also very highly expressed in steroidogenic cells, so cells that make steroid hormones. And so these include cells in the adrenal cortex, which make stress hormones. And it turns out that these cells take up a lot of cholesterol from HDL and store the cholesterol in lipid droplets and then use this cholesterol as a substrate to make steroid hormones. And when the HDL receptor is knocked out, these cells can no longer take up cholesterol. They become depleted of these cytoplasmic cholesterol droplets, and they don't make enough steroid hormones. So that this receptor plays an important role in the stress response as well. And then our interests, as I mentioned, currently are in this receptor mediating HDL signaling in cells like macrophages and in cardiomyocytes. So it seems to be playing an important role in a variety of aspects of HDL metabolism and HDL function. We're studying these aspects in mice, and the primary reason that we're using mice is because it's fairly easy to use genetics to try to tease out pathways in mice. And so I told you about the pomegranate extract experiment. We weren't really using a genetic approach to look at pathways in that experiment, but most of what we do involves looking at the effects of shutting genes off or turning genes on the development of atherosclerosis, coronary artery disease, heart attack formation in these models. And so we use mice because it's fairly standard and straightforward to generate lines of mice that have genes knocked out or genes overexpressed or knocked out or overexpressed either globally or in specific tissues. And a lot of people have been doing this, generating these mice over the last 30 or so years. And so there are a lot of different lines available. So we can take advantage of this to look at the effect of shutting genes off or turning genes on to discover pathways. So that's the primary reason for using the mouse as a model system. Now, a couple of things to note is that the standard mouse models for atherosclerosis, these are so-called ApoE knockout mice or LDL receptor knockout mice. They develop atherosclerosis in their aortas, but they don't develop atherosclerosis or at least they don't develop significant atherosclerosis in their coronary arteries. And so that means that their hearts are largely working normally, so they don't get heart attacks. In human disease, atherosclerosis in critical arteries like coronary arteries leads to heart attack. So we, in the process of studying this receptor, this scavenger receptor class B type 1 that I mentioned earlier, this HDL receptor, we knocked it out in mice, and we generated mice that lack the HDL receptor together with either apolipoprotein E, ApoE, or together with the LDL receptors. So in other words, we cross these HDL receptor knockouts to the standard mouse models of atherosclerosis. And our aim was to just see what effect the HDL receptor deficiency had on atherosclerosis. And what we saw, quite unexpectedly, was that these mice that lacked the HDL receptor now developed, in addition to more atherosclerosis in their aortas, they developed atherosclerosis in their coronary arteries. And the atherosclerosis in their coronary arteries was so severe that the mice developed heart attacks. And essentially all of the mice die fairly quickly after atherosclerosis is induced. So from the point of, how closely these mouse models mimic humans, the SRB1 knockout mice on various atherogenic backgrounds are kind of unique in that they develop atherosclerosis in coronary arteries. And as a result of this, the mice develop heart attacks. So that gives us an opportunity to study heart attacks resulting from coronary artery atherosclerosis, and to see whether we can, turn on or turn off pathways that affect the coronary disease, and how that affects how that affects the heart attack development. And that's really not feasible in the standard mouse models of atherosclerosis, which are the ApoE single knockout and the LDL receptor single knockout, because those mice develop athero in their aortas, but not in their coronary arteries. So from that point of view, the SRB1 knockouts, the HDL receptor knockouts, they kind of mimic more closely human disease in the sense that they develop atherosclerosis in the coronary arteries, leading to heart attacks. On the other hand, we have to realize that these mice are deficient in key genes in lipoprotein metabolism, and normal atherosclerotic disease in humans occurs in individuals who have more or less normal expression of genes in lipoprotein metabolism. So the mouse models are a bit extreme in that we've completely shut off expression of certain genes that we would never see completely shut off in humans. So we have to realize that as well, that they are a somewhat of a contrived model, because we have to go to extremes in order to induce atherosclerosis development in the mouse, as opposed to humans. So a normal run-of-the-mill wild-type mouse would not develop any atherosclerosis at all. So when I began, we used a technique called homologous recombination in embryonic stem cells. And essentially what this means is you generate a recombinant DNA that kind of matches the sequence of the gene that you want to target. So it matches the sequence on two ends of the recombinant DNA, but the middle is different. And what you do is you introduce that piece of DNA into embryonic stem cells in culture, and you rely on homologous recombination essentially by chance. So this is a very rare event. You have to screen through a lot of clones of the cells to try to find instances where homologous recombination has occurred. And what that means is that the two regions at either end of the DNA, the targeting DNA, match the specific regions on the gene, and that targeting DNA finds that gene and then swaps the content in the middle of that targeting construct for what's normally in the gene. So that's a very rare event. It involves screening of thousands of clones of embryonic stem cells to find one. And then once you find that, those embryonic stem cells then need to be grown up and then injected, microinjected into embryos at a certain stage called blastocyst stage. So this is when the embryo has divided as a collection of cells, but before any of the actual tissues or organs have really started to develop. There is a methodology called Cre-LoxP, conditional targeting. So that involves, sort of like I mentioned earlier, in generating a knockout mouse by homologous recombination, you can actually, instead of knocking out the gene outright, you can insert these short DNA sequences called LoxP sites around a critical segment of a gene, and then cross that mouse with a transgenic mouse that overexpresses a DNA recombinase called Cre, which is derived from bacteria. And so mice with overexpression of Cre recombinase in a specific tissue, for example, will have that gene knocked out in that tissue and only in that tissue. There are also some lines of Cre transgenic mice in which the Cre recombinase itself is not always active, but instead can become activated in the presence of a small molecule called tamoxifen, which is a derivative of estrogen. And so in these mice, the Cre recombinase only becomes active when the mice are administered with this tamoxifen molecule. And in the presence of tamoxifen, then the Cre recombinase can enter the nucleus, look for the genes that have these LoxP sites, and cut those genes out, and effectively knock out expression of the genes at a particular point in time. And so combining this with mice that have tissue-specific expression of this activatable Cre recombinase, you can knock genes out in specific tissues at specific times, and then test the effects on atherosclerosis development or reversal of atherosclerosis. So if we think that, a gene might be involved in migration of these inflammatory cells out of the atherosclerotic plaque, what we would want to do is allow atherosclerosis to develop. And then once the atherosclerotic plaques have developed, then introduce the small molecule tamoxifen to activate the Cre recombinase to shut off expression of that gene and see if that allows the macrophages to move out of the atherosclerotic plaque. So those kinds of experiments are possible with these conditional knockouts. Probably the most important direction for this is really trying to understand pathways and how to alter pathways to potentially reverse atherosclerosis development. So we know that, the artery wall thickens. It's because of an influx of cells into the artery wall. And there's some evidence in animal models that perhaps this process can be reversed. And if we can identify small molecules that might act as drugs that trigger that reversal pathway, then we can move those into larger animal studies, so out of the mouse into larger animal studies, and potentially move those into studies of non-human primates, and eventually, if they pass those into clinical studies. I think the reversal is going to be a major breakthrough if we can get that working. And the reason for that is, essentially, if once an atherosclerotic plaque develops and an artery is narrowed, it stays narrowed, then all we can hope to do is sort of stabilize the disease. But if we can actually open that artery back up non-surgically with a small molecule, with a drug, then, I think that would be a major breakthrough. I think that healthy lifestyle choices are really critical, and that people tend not to think about this until they've actually developed the disease or had a heart attack. But this is something that people really should develop from an early age. Healthy eating habits, healthy lifestyle habits, exercise, and the earlier you start and the more you maintain it and longer you maintain it, the reduced risk of developing this disease later on. So I think that this is something that is important, and I think that a lot of people would be much healthier in general if they followed this. Lifestyle is perhaps one of the best ways of preventing atherosclerosis, but it's perhaps one of the hardest to implement, I think, on a public health basis, because people don't like to change their habits. So certainly there are opportunities for undergraduate students to become involved in research. My advice is to reach out with professors who are running research labs to ask about opportunities in their labs. A lot of the undergraduate programs, for example, the Biochemistry program or Biomedical Discovery Commercialization program, BHSC, a Bachelor of Health Sciences program, chemical biology, you name it, they have opportunities for undergraduate students to do research placements for credit in faculty members' labs. So those are clearly one avenue to get involved in research. The advice that I would have is, reach out to faculty members early to try to arrange for a placement in their lab, because there is quite a lot of competition. So there are opportunities through your undergraduate programs, but there are a lot of students who are vying for these positions. So, cast a wide net, reach out to as many faculty as you can, and don't restrict your reaching out to just faculty at the main campus. Realize that there are also McMaster faculty at all of the various hospitals around the city, and who have labs at the hospitals. My own lab is located on the campus of the Hamilton General Hospital, as opposed to the main campus. So, you should reach out to all of those faculty as well. So cast a wide net. because there is quite a lot of competition for these spots.